where the life and death decision has already been made.
The rush to get fundable lines onto the presidential list before the 2001 deadline produced some questionable results.
So many of the so-called stem cell lines that were submitted at the time were actually not lines. They couldn't self-renew, which is the hallmark of the stem cell.
With federal funding out of the picture, most researchers, like Dr. Susan Fisher, were left with no options. Fisher's work on human placental cells would require the development of new stem cell lines.
I think there are a number of quite good cells on the registry, but also when you want to encompass all of human biology and cell lines, you can imagine that less than 20 is not going to do it.
This is where the state of California stepped in with a radical new solution. Put it to the voters.
59% of Californians voted in November of 2004 to commit $3 billion over a period of 10 years to stem cell research. This, in a way, positions California to fill in the areas that the federal government cannot venture at this moment.
The $3 billion provided by Proposition 71 established the California Institute for Regenerative Medicine, or CIRM. Its mandate is to evaluate proposals and distribute funds to universities and other research institutions. The new law triggered a gold rush of stem cell science. For world-class researchers like Dr. Deepak Srivastava, Prop 71 finally made new research possible.
I was interested from the time I was in medical school about a very basic question about how a cell decides what it's going to be. And as I went along in my medical training and became a pediatric cardiologist, it became clear that the clinical problems I was addressing were in fact a problem of a stem cell, early embryonic stem cell, not being instructed to become a heart cell, or subsequently not positioning itself properly with its neighbors to form the proper three-dimensional structure of a heart.
Srivastava and his team hope that by carefully controlling early stem cell differentiation, they can create cultures of partially developed heart stem cells called progenitors.
If you introduce a stem cell into an organ, then it's absolutely critical that it only become the cell type of that organ and nothing else. So you want to introduce a cell that can still expand and divide once you put it in, but it's already far along enough on the way that it's only going to become, say, a heart cell or a brain cell, etc. You don't want a brain cell in your heart.
These progenitor cells would act as the raw material to replace damaged heart tissue or even entire heart structures.
One can imagine growing on a scaffold, what we call biodegradable scaffold, where a structure of a valve is created with a matrix, and then it's seeded with stem cells that can turn into the covering layer. And then once the structure is there, the inside scaffold can dissolve away and you're left with the outside cells that came from the same patient. That valve tissue then could be transplanted, right now implanted, back into the patients for use.
Research into treatments such as this would have been difficult, if not impossible, without the alternative funding source offered by the California Institute of Regenerative Medicine.
No single person or single agency is smart enough to think of where research will go, where the most promising avenues and where the new breakthroughs will come from. Therefore, it's really important to cast a wide net, and these initial initiatives tried to do that.
Science is all about innovation, creativity, and freedom of investigation. The last several years, that's been somewhat stifled, at least in the stem cell field. Prop 71, I think, has allowed investigators like myself to think freely about how we can move human embryonic stem cell research forward without having to compromise on pursuing the right scientific question.
Eventually, scientists say, stem cells may one day provide cures for Parkinson's, juvenile diabetes, and many other diseases. And in California, the seedbed for this work has been prepared. For Dr. Srivastava, It's a medical frontier that holds unimaginable promise.
The most exciting thing to me about the research is the fundamental question of life. How it is that a cell that starts off with one type of DNA can turn into the 200 or so different types of cells in the body. It's the excitement of that discovery that will form the foundation for all the therapies.
So, did you find that useful? Was that a useful video kind of bringing you up to date on this field? And that was done not terribly long ago, within the last year. And you know, Shrivastava said something very profound. What is life? When's life begin? And you know, evolutionarily, it's taken millions of years, correct? That's not why you're here tonight, because we don't believe that by faith. That ultimately life occurs at conception, and we're created in the image of God. So the question is, what are stem cells? And is there a difference between embryonic stem cells and adult stem cells? And the obvious answer is yes. And we're going to get into that in more detail tonight, because that's a very important issue. Keep that in mind going forward here. What is the potential value of stem cells in medicine? You got a hint of that from the video, and we'll talk a little bit more about the potential applications. And how do stem cells fit with biblical truth and principles? And that's the question I presume we're all interested in answering if we're going to be here tonight. So stem cells basically are unique cells that can divide and make identical copies for an indefinite period of time. This is a concept called self-renewal. Most cells, one of the first projects I did as a research fellow when I came out of my medical training, I was at the University of Washington at the time, and what we were studying is skin fibroblasts. And skin fibroblasts are cells, if we biopsy the skin and we put the cells in a dish, the fibroblasts are the predominant cell.